So that gets us to think about all of these phage genomes, the sequences and the sizes that I've showed you, and I've given you some sense of how the genomes can be organized. That brings us to the question as to how similar or different are these at the genetic level. And I think a good place to start is by thinking about how they appear if you take these known genome sequences and compare them at the nucleotide sequence level as just a general measure of genetic diversity. And perhaps before I do that, I'll just comment that you do have to be very careful about the point I made here about these genomes having similar organizations and a similar morphotype relative to this guy down here that has a rather different organization and is a different morphotype. And the reason why you have to be careful is because you can identify lots of genomes of this type, for example, which are these cyphoviridal types with these long non-contractile tails. But we know that if you ask the question, does this morphology, does the way they look in the electron microscope, does this reflect their genetic similarity? The answer is, generally speaking, no. These are not necessarily genetically similar just because they're morphologically similar. In fact, it's quite common to find, and I'll elaborate in a minute when we look at the nucleotide sequence comparisons, but it's quite common to find two phages that may look essentially identical in the electron microscope, but have no genes and no DNA sequences in common at all. So we have to be very careful about how we interpret morphology in terms of evolutionary relationships, and emphasizes that we really do need to look at nucleotide sequence and genetic relationships. So first of all, broadly speaking, and I won't show you any of these plots because they would be very boring, that if you look at and compare nucleotide sequences of phages that infect different hosts, the nucleotide sequences tend to be very different from each other. So at the broad level, and we think about all of those 495 phages, the genome sequences are very different. We can go from there and we can narrow in and ask, well, how similar are the genomes that infect any one common host? And now that more genome sequences are becoming available, that's a question that you can ask for E. coli phages, the so-called dairy phages, many, many other different types of phages. And I've just shown you here a couple of studies, one looking at the phages that infect, or a group of phages that infect Staphylococcal aureus, which includes the original Twor phage from Frederick Twor, and then a group of phages of Pseudomonas aeruginosa. And so what is being done here is that the complete genome sequences have been determined. They've been joined end to end for all of these staphylococcal phages here, and then compared against each other. And you can see a diagonal line that represents the identity of phages with themselves, here and here again. And then within each of these triangular areas here, you can see that where two genomes have clear nucleotide sequence similarity, it is illustrated by these diagonal lines. So here, for the staphylococcal phages and Pseudomonas phages, you can see there are sets of genomes here, a cluster that are related to each other, but not to these. There appears to be a rather larger cluster here, some of which are quite closely related to each other, and some less so. Another group here, and a couple of stragglers that really aren't related to anything. And a similar picture emerges for these Pseudomonas phages. There are some that are related to each other, but even so, there's a relatively high level of genetic diversity. How the phages differ in different hosts really depends on which host you look at. You can see that these appear to be more genetically diverse than these, but I think while we're looking at just 20 or 30 genomes, it's still a relatively small number. And clearly, these represent relatively small subsets of the populations out there. We have by no means saturated the available genome sequences in these displays, and it emphasizes the need to expand this type of analysis so that we can understand these questions of genetic diversity. This is just looking at mycobacteriophages, 50-something odd genomes here that are compared by the same technique, just linking together the nucleotide sequences and then comparing them. And here you can see that we've begun to put names on these clusters of genomes that appear to be more closely related to each other than they are to everything else. But I should emphasize that once again, just as you saw perhaps in the Pseudomonas phages, whilst there are clusters that are emerging, there are nonetheless quite diverse in terms of their nucleotide sequences. These are not simply 50 variants of a common nucleotide sequence. They're quite different to each other, but they're not universally different to each other. So that's true for the mycobacteriophages, it's true for the other two and different groups of bacteriophages that you look at, but the extent of the diversity appears to be different from host to host. And as I emphasize, in each case, I think the numbers really are relatively small, and one has to be very careful about extrapolating what we see from this analysis to the broader population. If we had 100, 200, 500, 5,000 genomes, it may or may not resemble this degree of similarity. We just don't know. So given those concepts and those thoughts about how extensive the genetic diversity is, the question becomes as to how related are these genomes, the genomes that are encoded within phages that infect a common host or even different hosts. And if we begin to look at the sequence relatedness, not at the nucleotide level that I just showed you, but by comparing the predicted amino acid sequences of the products, it gives us a much more sensitive level of comparison, and in principle enables us to identify evolutionary relationships which are much more distant, sufficiently distant that you've lost the nucleotide sequence comparison that's identifiable, but can still see the relationship by conservation of the amino acid sequences of the predicted proteins. And so this game of making these comparisons is something that can be done with all bacteriophages for all different hosts, and I'll just tell you a little bit about the lessons that we've learned from looking at mycobacteriophages. And so this simply reminds me to show you that one of my favorite bacteriophages, L5, which was the first mycobacteriophage genome that was sequenced, and I think the first double-strand, large or medium-sized double-strand DNA tail phage outside lambda and T7. That the genes can be identified as these boxes, and so by identifying and annotating the genes, or the putative genes, it provides to us the amino acid sequences that we can then use for comparison purposes. And when there was only relatively few genome sequences available, one could simply ask who was related to whom and express these relatives, these relationships, on this kind of genome map. So when one determines a new bacteriophage genome sequence, you can bioinformatically predict where the genes are and then collect essentially a list of amino acid sequences that can be then used for comparison purposes using programs like BLAST and Clustal, etc.